Hello, everyone. This is Yufeng. I'm from University of Connecticut. Today, I'm going to talk about the inference of a population admixture network. Demographic history is a central subject in population genetics, which has a model different type of processes such as population divergence, population size changes, and admixture. Admixture refers to forming a new population from two or multiple source populations by mixing the genes together to form a new population. Let's see the demographic history for human in the past 600,000 years. This is from Wikipedia, and the y-axis here are the time. The horizontal axis is a geographic location, say Eurasia and different parts of Africa. And from this figure, we can see the admixture, the forming of new population from two or more source population, kind of a fairly common. Say there's some admixture here, right, in the ancient part of the history, and also there are more recent admixture. Right, one involving Neanderthal of Homo sapien, which has been reported widely in literature. So where we like to do some inference of the admixtures from the genetic data. This is because the demographic history is not directly observable usually. To do such kind of inference within the model, simplify the model. Here we consider this model called admixture network model, which only model the population divergence and the admixture. It doesn't model more complex situations like population size changes within the same branch and also other type of complex migration. The figure here shows there are this four populations A, B, C, D, time flow from top down, and note here the important thing to note is there's an admixture here, form the population C. So C is admixed. So this is the main model we are going to consider in this talk. Now let me tell you exactly what type of data I'm going to use for inference of the admixture network. Here I mainly use the haplotypes, which are the linked alleles and the multiple single nucleotide polymorphism, or SNPs. SNPs I assume to be binary or 0, 1. So you can see the figure on the left. There refers to haplotypes. Here each row is a haplotype, and each column is a SNP. So there's 7 SNPs and 8 haplotypes in total. Here, these haplotypes are from 4 populations, A, B, C, D. So each population has 2 haplotypes. So A has A1, A2, and so on. And I break the haplotypes into 2 loci. So the first loci has 4 SNPs, and second loci has 3 SNPs. The admixture network inference is a challenging problem in computation. Due to private communications with from Lou Xinzhang, I was told that for 10 populations with 2 admixture events, there's up to almost like a 300 billion possible networks. So this is very, very large, and fortunately, Lou Xin told me only after I finish this work. Otherwise, I may be frightened by this number and not even get started. So the current method for inferring the admixture networks from genetic data are based on the single SNPs. That is, they tend to treat each SNPs independently from each other. The most popular method is called tree-mix. It's by Pickrell and Pritchard. There's also another method called mix mapper. These methods are fast, but the downside is that they lose the important linkage disequilibrium information contained in the haplotype, but not in the individual SNPs. In this talk, I will describe a new method called GTMX. This is based on haplotype, not single SNPs. The GTMX method is slower, but experience shows that it's more accurate than the existing method like a tree-mix, even when GTMX is only given smaller data than the existing methods. Let me tell you the overall ideas of GTMX. GTMX is a maximum likelihood approach, inference approach. The most straightforward way to do MLE is to find the network that maximizes the likelihood of hypertypes, which is given. The challenge, however, is that the likelihood of a hypertype is well known to be a very hard problem in population genetics. So to get around this problem, GTMX first infers the local gene genealogies of each locus from the hypertypes. So here, the gene genealogy refers to the evolutionary history relating the population hypertypes. And then, with this inferred local gene genealogies, GTMX find the network that is maximize the likelihood of these gene genealogies rather than the likelihood of the hypertypes. The main reason is that the likelihood of the genealogies turns out to be easier to compute than the likelihood of the hypertypes. And also, that gene genealogies remain informative for admixture. It contains important LD or linkage disk equilibrium information, which is not captured by single SNPs. The challenge is that the inferred gene genealogies tend to be noisy, and also computing likelihood of gene genealogies still not trivial. In this talk, I will show GTMX can perform accurate inference network despite these challenges. Now let me tell a little bit about gene genealogy inference. This is a hard problem, but has been studied actively recently. Gene genealogies are strongly influenced by recombination, which is illustrated here, which take a prefix of one sequence and take a suffix another sequence and merge together to form a new sequence. Let's consider this figure for as a plausible gene genealogies for 4 sequences 0, 0, 0, 1, 1, 1, and 1, 0. And let's look at this genealogy. And the important thing here is the recombination. Let's trace the history on the first position, right? Due to recombination, it first goes to the left, right? And then it forms a genealogy like this, right? So this gave one plausible gene genealogies here. Now let's look at the next position. So here, this position, we trace back, and then due to recombination, this time we'll go to the right. So we have a history like this. So this gave a different gene genealogy. Right. So infer local gene genealogies from haplotype is a challenging problem, and the first practical approach is called algorithm, which is a probabilistic inference, but fairly slow. Several years ago, I developed a method called RUNT+ with my student, which improved my earlier method called RUNT. Since then, there are 2 methods being published, which both appear in Nature Genetics. In this talk, I will use RUNT+. This is not just because RUNT+ is my own method, really because RUNT+ is easy to use, and you do need to provide population genetic parameters such as recombination fraction and also work reasonably fast. What does the inferred local genealogies tell us about the admixture? Let's consider one possible gene genealogies inferred in one locus on here. So let's look at this part. This shows that the lineages from population B and C tend to coalesce. This is an indication that population B and C are closely related. Let's look at another inferred gene genealogy, another locus. And here, we look at this part. It shows that lineages from population C and D tend to coalesce, and that means C and D are closely related. And this indicates that population C is closely related to both population A and B, which means that C is likely to be admixed. However, the real situation is not as always this easy. First, genealogy inferred tend to be very noisy, and also there are also complexity caused by the stochastic nature of a coalescent. GTMX approaches combine information from gene genealogies T1, T2 from multiple loci for inference of admixture. And the basic idea is that each genealogy tells something about admixture, and then this something is measured by this probability. And then we combining this into the likelihood computation of the gene genealogies. So how to compute this likelihood? For now, let's assume there's no admixture. So there's a single population tree. It turns out computing the Gentry probability on a given population tree itself is not trivial. The gene tree probability is the probability of observing a gene tree topology T for given population tree TP due to the coalescent. There's no known polynomial algorithm for this problem. The first algorithm is due to 2005. Then I developed an earlier algorithm called STELS in 2012, which is much faster than the original algorithm. And the STELS 2 was published a few years earlier. This is a heuristic version of the original STELS algorithm, and it's much faster and seems reasonably accurate. GTMX uses the STELS 2 algorithm for better scalability of the inference. So what about the probability of gene tree on a network rather than a population tree? Computing the probability of a gene tree probability on a network turns out to be much more challenging in computation. GTMX apply a heuristic to make this computation more practical for inference. The critical observation here is that the network contains multiple population trees. So let's look at this example here. If we consider the where admixture occurred, we can have a one population tree by taking this branch of the history, and which we have a TN1, and we can also get another one we call TN2 by taking another direction. So this go to two population trees from this network. And the critical assumption made by GTMX is that the probability of is a weighted sum of the probability of a genealogy on each contained population trees. The weight is computed by the so-called admixture proportions in the network. So here is a simple calculation. It's a weighted sum of the gene tree topology T on two contained population tree TN1, TN2, right? And this is heuristic, not exact, but it turns out works reasonably well. With likelihood computed, now the remaining task is to find the maximum likelihood admixture network. The first step is initialization. We need to find a good initial network from the gene genealogy. And then GTMX perform hill climbing by a local search. That is, move on to the next network nearby if the network is a better likelihood than the current one. So to do this, we need to optimize branch lengths, just like the maximum likelihood phylogeny inference. And we also need to find the nearby networks. We perform so-called nearest neighbor interchange, NNI, to find the network from the current network. Practical issues contains the first efficiency. There may be too many inferred gene genealogies, and GTMX implement effective tree picking procedure. See the paper for more detail. And GTMX also need to find the initial network which using a heuristic. This turns out to be an important step. See the paper for more details. Now I show some simulation results. First, I'm going to vary the number of populations from 4 to 10. Here I am. So there's a number of population from 4 to 10, and I'm going to evaluate on the inference error. So this is the inference error. The smaller the better. Here I run GTMX using only small data, which is contain 4 haplotypes per population. And you can see from here, the inference error of GTMX is actually the lowest among all we compared. The next better one is TRIMIX. TRIMIX is run with much larger data with 100 haplotypes per population. That is 25 times more than used by GTMX. And you can see, even so, TRIMIX not performing as well as GTMX. So the next step, I evaluate the varying number of haplotypes per population. I fix 6 populations, and I use from 2 to 100 haplotypes per population. Due to competition efficiency, GTMX only run for up to 8 haplotypes. And you can see here with GTMX is here. GTMX results with 8 haplotypes per population is actually perform better than TRIMIX with 100 haplotypes per population. So this indicates that GTMX my method is actually more accurate with small data than the existing method like TreeMix. Even the later is given much larger data, like 25 times as large, even though GTMX is usually slower than TreeMix. We now demonstrate how GTMX works on real data. This is a 1,000 genome data, which pick 10 populations, 2 African, 2 East Asian, 2 European, 2 South Asian, and 2 admixed Americans, Mexican, and Puerto Rico. I run GTMX with haplotypes from 50 evenly distributed loci from the first 10 chromosomes. I use 2 diploid individuals, that is 4 haplotypes per population. I don't use outer group. And you can see here, first of all, the 2 populations from each superpopulation, African, European, and so on, they are grouped together, which is as expected. And also you can see the admixture here are mainly affecting the admixed American population, Mexican and Puerto Rico. So this is matching what we expect. So even though the true network is unknown, and this shows that GTMX actually can work on the real data. Now I compare TreeMix on 1,000 genome project data. I first use EvolCompGen and African population as the outgroup. The results are shown on the left. Just as before, the 2 populations from same superpopulation tend to cluster together, and also the 2 admixture events affecting the American populations also as expected. Here I run TreeMix with whole genome data and with 20 individuals. That is 40 haplotypes per population, which is much larger than that is given to GTMX. The problem I observe is that, however, when I run TreeMix with no outgroup, the results, even though you still have the 2 populations clustered together on the same superpopulation, but the admixture doesn't quite make sense, and also the routing seems mess up. You probably know the so-called Auto-African history. So this indicates that TreeMix apparently doesn't work quite well when there is no working outgroup. Now summary. In this talk, I described the approach called GTMX, which infer admixture network from haplotypes, not single SNPs. Results shows that GTMX, our new method, outperform existing methods, even slower, but it's more accurate than the existing method.